Hello, everyone, and thank you for joining us today. I'm Sean Fensky, Editor-in-Chief of Medical Product Outsourcing and moderator for today's program. You've logged on to the MPO webinar, What Makes a Successful ISO 18562 Toxicological Risk Assessment? Before we get started, I have a couple of housekeeping announcements. For those of you who are interested in the information you hear today, once the webinar has ended, or if you'd like to forward it to a colleague, this session will be archived for a year. The archive will be able to be accessed through the MPO website, which is at mpomag.com. Also, we will be accepting questions throughout the presentation. You can type your question into the box provided in the webcast window. We'll collect all questions and hold them until the end, but please feel free to type in questions as you have them. And now, before I introduce today's speaker, I'd like to take this opportunity to thank our sponsor, Nelson Labs. Nelson Labs is a global leader in microbiological and analytical chemistry testing and advisory services for the medical device and pharmaceutical industries. The company serves over 3,500 customers across 13 facilities in the United States, Mexico, Asia, and Europe. Nelson has a comprehensive array of over 800 laboratory tests supporting customers from initial product development and sterilization validation through regulatory approval and ongoing product testing for sterility, safety, and quality assurance. And now let's get started. Today we're joined by Nathan Frederick, toxicologist for Nelson Labs Expert Advisory Services. As part of the Expert Advisory Services team, Nathan uses his chemistry and biology background to evaluate extractables and leachables through toxicological risk assessments. In addition, he specializes in evaluating air volatile organic compounds from gas pathway devices per the ISO 18562 series of standards. Nathan has also worked with the Nelson Labs Antimicrobial and Filtrations Department as a study director who oversaw filter sterilization validations. Now we'll begin with today's presentation. Nathan?
Hi, yes, thank you for that introduction, Sean, and thank you for having me today. As mentioned, my name is Nate Frederick. I am a toxicologist with Nelson Laboratories. I currently write toxicological risk assessments from chemistry testing of medical devices, and I help leverage the biocompatibility safety for end users of said devices. Thank you once again for joining me today as we'll be taking a deeper look into the ISO 18562 series alongside the toxicological risk assessments to follow in my webinar, What Makes a Successful ISO 18562 Toxicological Risk Assessment. Over the previous decades, the ISO 10993 International Standard Series has been the overarching set of standards that helps provide guidance on how to evaluate medical devices for biocompatibility. Many documents have evolved, and they continue to evolve. As such, the new ISO 10993-17 release is very anticipated. However, there has been a split in these overarching documents because of special cases of externally communicating devices, such as those that carry air or gas to the lungs. When a gas is the carrier of an externally communicating device, the risk profile is a bit different than if the carrier was a liquid, which the liquid is the suggested matrices of the ISO 10993 series. From a toxicological standpoint, more often than not, these gases, when exposed to the lungs, are more toxic, problematic, and in some cases, unsafe exposures to the chemicals that could come off of medical devices during their intended use. To see how these standards are a bit different, the 10993 suite are the standards that cover all biological evaluation of medical devices. However, the ISO 18562 series looks at the gas pathway within healthcare applications. The use of these two standards goes hand in hand, and they go hand in hand with the various FDA guidances to further support the safety of all medical devices. As an introduction to ISO 18562, the main goal of this series is intended to be a guidance to determine the potential biological risks, such as looking at the risk of the materials and processes to the patient. For the risk management stance, ISO 18562-1 is responsible for the overarching risk management process of these potential indirect externally communicating devices. While the categorization is something that most of us are familiar with, it won't hurt to briefly touch on it, so we're all on the same page. The term indirect contact is used for a device or the device components in which a fluid or gas pass through before they come into physical contact with the body or body tissues. We can usually look at this as the device component does not physically contact the body or tissue. Furthermore, according to ISO 10993-1, this indirect tissue contacting will lead to insufficient assessment and testing of these gas pathway devices. This is really where ISO 18562 becomes the star player. As a further introduction to the 18562 series that will be applicable within this webinar, we have ISO 18562-1, the evaluation and testing within a risk management process, -2, testing for the emissions of particulate matter, -3, tests for the emissions of volatile organic compounds, which will often be referred to as VOCs. And then we have -4, testing for leachables and condensate. I really should have put an asterisk by this, or simply have just changed this to ISO 10993-18 for FDA submissions, but I'll touch on that shortly. Furthermore, the FDA fully accepts 18562-2 and 18562-3, and it partially accepts -1 and -4. In -1, the FDA omits the irritation testing, and in -4, the FDA omits irritation testing and the worst case patient contact of 1 ml of condensate per day. They state there is not enough data to support this condensate dose. In lieu of -4, ISO 10993-18 may be a substitute for the FDA submissions. This is where a 72-hour extraction of the device while it is fully submerged, and then a full chemistry panel is ran on the extracts. All of the compounds seen in the extracts can be assumed to reach the patient, but this is a gross overestimation. And then I'll touch back on the practicality of this further on in the webinar. As an outline for what makes a successful ISO 18562 toxicological risk assessment, we could use this pyramid, for example. On the bottom, we have robust testing, as the base of the pyramid will need to be the strongest part before you can continue to build. Here is where the medical device goes through all of the applicable testing. This is the most important part, as if the testing does not meet clinically relevant, there is really no point to run a toxicological risk assessment on the results. The next level is a reasonable interpretation of results, or relevant toxicological analysis. This level is where the chemicals are evaluated, tolerable intake, tolerable exposure, and margin of safety values are calculated. They could be derived from literature. Then lastly, on top of the pyramid, if any risks are identified, they could be mitigated appropriately, or further testing may just be required. This level is all about appropriate risk mitigation. Our whole end goal is patient safety, and mitigating risk requires special care and expertise. This pyramid sums up what makes a successful 18562 toxicological risk assessment. So really, without diving into any specifics, my whole webinar can be summed up in this slide. But we do want to get into the specifics. So right away, starting with the robust testing, we first have the test selection. Here, we start to build the base of our pyramid. If we have a dry air pathway, we'll just be looking at running a particulate test via 18562-2 and a VOC test via 18562-3. However, if there is a chance that humidified air can be transmitted through the gas pathway, we will additionally look at condensate testing through -4 or ISO 10993-18 for the FDA submissions. If no surface contact testing is performed on the device, then the big three, cytotoxicity, sensitization, and irritation, will need to be tested. All of this testing should be performed on a sterile, final finished product that has gone through all of the applicable sterilization and packaging steps. There are some cases where R&D can be done on the device that's not in its final finished form. That's why it's R&D. However, when submitting to regulators, it is expected that the final finished product be tested for the submission. As mentioned previously, particulates must be tested for all air pathway devices. There are three options: a single particle filter, inertial particle separator, or a particle counter. These particulates are tested by air being passed through, or through and around the device, and then being collected and ultimately counted. This air must be of a certain flow rate and temperature. For particulate testing, it requires the highest clinically relevant flow rate or the highest flow rate for which the medical device may be set at. This high flow rate must be used when sampling, as it is seen as a more worst case scenario. Of this air that has been sampled, particulate matter 2.5 and particulate matter 10 is then analyzed and taken into account within the toxicological risk assessment. Per the US EPA 40 CFR Part 50, the limits for the respective particulate matter size, excluding the nanoparticles, are as follows. 12 microgram per meter cube for the 2.5 micron particulate matter, and 150 microgram per meter cubed for the 10 micron particulate matter. From toxicological standpoint, size matters. The smaller particulates are more toxic than the larger ones for multiple reasons. The 2.5 micron particulates can be suspended in air for longer periods, thus increasing the chances of them getting breathed in. If they have been inhaled, they can penetrate deep into the lungs alveoli. This is where the gas exchange happens, and from here, there's a possibility they can enter the bloodstream. I'm not going to get into all the nitty-gritty details of the mechanism of action. However, health effects from particulates can range from development of asthma and bronchitis to heart attacks and strokes. Moving on to volatile organic compounds, this test is required for all gas pathway devices. Here, the devices are tested in a similar fashion. Clean, dry air, or dry nitrogen may be pushed through the device. If the air is tested just through the device, it's only testing the interior of the device. Or the devices can be sampled by pushing air through and around, testing both the interior and exterior of the device. This flow rate, however, differs from particulate testing, as we need to look at the lowest clinically relevant flow rate. If, for example, the lowest flow rate of the device is 0.5 liters per minute, it may not be a bad idea to test at 0.1 liters per minute, just as long as it's below that 0.5. The method that the FDA has requested gas pathway medical devices to be tested to is the TO15 method, or it could also be referred to as the EPA canister method, which Nelson Labs has adopted and tests to till today. In this method, the air samples are collected in a canister that are made out of stainless steel, as pictured. Once collected, air is then analyzed using the GCMS, which then identifies and quantifies the analytes seen. We are left with an extensive list of quantitative compounds and semi-quantitative compounds. The length of time for this VOC sampling should be multiple 24-hour periods. It is typical for sampling to occur in 2 separate 24-hour periods, even for long-term devices. This gives us 2 days worth of data, the first 24 hours and the second 24 hours, 24 to 48. Here, we can look into a little bit of the release kinetics and see if compounds go down on day 2 when compared to day 1. If there are expected to be a lot of volatiles come off the device, it may be a benefit to increase sampling out to 3 or even 4 days. Additionally, if your specific device, it may need to be tested for inorganic gases, such as ozone, carbon monoxide, or carbon dioxide. Some guidance is provided if your device has electromechanical or electrostatic parts, or just if inorganic gases could obviously be generated. For a device to generate ozone or other inorganic gases, although it is uncommon, but it's not unheard of, there's a few devices that come to mind. The devices at risk are any devices that are intended to perform a chemical reaction. For example, an N2O generating device, or a device with an active disinfecting element. For example, if UV lights are a part of the device and they're intended to disinfect. Then there's some devices with lower risk, but they are still possible to be questioned. These are devices with motors in the gas pathway. If the gas a patient breathes goes through an electric motor or an electric discharge from the motor, it could produce ozone. And then lower risk, but still within the realm, is any device just with moving parts in general. So I quickly ran through the various types of testing, but here's a quick schematic of how testing is essentially set up. We have dry air coming in, and then it is filtered through a carbon filter. The air is then sampled for a blank, and in turn continues to go through a flow meter, where the adequate flow and temperature are monitored. After it goes through the flow meter, it then comes into contact with the test article. Once again, the air may go through the test article, only testing the inside of the device, or it can go through and around the device, testing the interior and exterior surfaces of the device. The test article can be manipulated in the chamber, and it can either or can be set up. From here, the air is collected into the sample canister, and additional air is vented. The sample canister is then ready for GC-MS analysis. Once the gas has been analyzed, it is then blank corrected, and results are reported in a concentration of microgram per meter cubed. These results are split between the two sampling days, the first 0 to 24 hours and the second 24 to 48 hours. Since our results are in a concentration, we can easily calculate the patient exposure. This is done by using the maximum amount detected between the two 24-hour periods, multiplied by the breathing rate as outlined in 18562. The breathing volume for adults is 20 meters cubed per day. For pediatrics, it is 10, infants is 2, and neonates is 0.21 meters cubed. For the sake of the toxicological risk assessment, the daily amount from the highest sampling period is expected to be exposed to the patient for the entire lifetime or use time of the device. As mentioned previously, condensate may be formed in the device and can make its way to the patient. This condensate has the potential to extract chemicals from the gas pathway of indirect contacting devices. If there is the possibility of condensate, whether that be from the device itself or the patient's rebreathed air, an extractable study must be carried out. ISO 18562-4, it is not recognized by the FDA as previously mentioned. So a substitute is to run a full extractable and leachable panel with the guidance of 10993-18. Additionally, the testing approach may be verified through a biological evaluation plan, which can also be done with the expert advisory services here at Nelson Labs. These extraction conditions, as outlined in 10993-18, are where the device is extracted by being fully submerged, much like this hand and this beaker. Devices are normally extracted for 72 hours at 50 degrees Celsius. Mostly, the devices are extracted in water. However, if a patient's rebreathed air or if there are drug excipients that may come into contact with the device, ethanol, IPA, or hexane may also be used as an additional extraction solvent, if they're compatible. When we perform this extractable and leachable study, we are essentially casting a very big net, looking for a large variety of compounds within the extract. The substances we could be identifying are either inorganic or organic based. Our ICP-MS or ICP-OES will pick up any metals, while our GC-MS will pick up any volatile organics or semi-volatile organics. And then lastly, we can identify any non-volatile organic compounds with LC-MS. While that was a lot of information in a very quick amount of time, we must keep in mind that robust testing does build our base for our pyramid, and everything else will compound on top of it. Very quickly, these are the key things that should be remembered as far as testing goes. The worst case clinical conditions must be considered. A high flow rate for particulates. We want to blow out all that residual manufacturing dust, capture and analyze it. A low flow rate and high temps for the VOCs. And inorganics if there's any electromechanical or just mechanical parts in general. If condensate can be formed and reach the patient, extractable and leachables must be considered using ISO 10993-18. And all of this testing can be done at our Nelson Labs locations. Once we have generated results and see what compounds or analytes come off of your medical device, we can then assess everything all at once within a toxicological risk assessment. This is the beginning to the second level of the pyramid. We want a relevant toxicological analysis. However, I must emphasize on relevant, as our end goal is patient safety. Within a toxicological risk assessment, we include device description and categorization, explanation of testing, and then a toxicological assessment of all the analytes seen. We can assess particulates, VOCs, inorganics, and condensate from E&L all within a singular TRA. Here, we can derive various tolerable exposures, tolerable intakes, ultimately providing a margin of safety value. We then have a short calculation discussion, and if there are any compounds that are known to have adverse toxicological effects, we can provide full multi-page write-ups. If a toxicological safety is not supported, we may move on to the top of the pyramid, risk mitigation. However, risk mitigation comes with regulatory risks and it must be handled appropriately. The basic steps for a toxicological risk assessment are generate your chemistry results from your medical device. This is done with that robust testing. Then research available toxicological data for each compound from literature reviews. Here we try to determine the NOEL, the no adverse effect level, the low L, the lowest adverse effect level, or any other relevant point of departures. These are then computed into a tolerable intake and a tolerable exposure, taking into account uncertainty factors throughout. And then it is further calculated into a margin of safety. These point of departures are researched through a literature review of trusted databases, such as PubMed, ECA, ToxNet, ATSDR, and the IRIS database. Let's jump back into that data set that was presented earlier as the quantitative results from the air VOC sampling. Before I get into these results, I quickly want to touch on the threshold of toxicological concern. It's also known as the TTC. The TTC concept was developed as an approach to define an acceptable or safe intake for any chemical that does not have any relevant toxicological information. These thresholds vary based on the clinical device use as seen here. The TTC is a very conservative and it takes into account the most sensitive toxicological endpoints. Any compound below the TTC would not pose a risk to the end user. Real quick, since we have this data set here, let's use this as a quick case study. Say this data is from a long-term device that will be used in adult patients only. We'll be looking at the TTC of 40 microgram per day. Keep in mind, this data is for adults and is the maximum detected between the 2 days multiplied by the breathing rate. That leaves us with 4 compounds below the TTC and 4 compounds above the TTC. For the compounds that are above 40, we have to derive a TE and margin of safety value for each one. A safe margin of safety value would be anything above 1. And if there happens to be a margin of safety value below 1, we could attempt that risk mitigation. Additionally, it seems as if regulatory bodies have changed their stance on the use of the threshold of toxicological concern. If the compound has relevant toxicological data, we are expected to derive a TE and MOS for each one of these compounds. As seen here, these 4 compounds are all below the TTC, but there is that relevant toxicological data, and we will derive a TE and margin of safety for each compound. The margin of safety is essentially the end goal for each compound. This is a way to see if the said compound is safe. To calculate the margin of safety accurately, we start off by deriving the tolerable intake. Here, the NOEL, point of departure, which is derived from literature, is divided by the various uncertainty factors. These uncertainty factors could be different each time a TI is derived, as these are based on the inter-individual variations between humans, the extrapolation between humans and the animal that was used in the toxicological study, and the quality and relevance of the experimental data. Additional uncertainty factors can be added as the toxicologist sees fit. After taking these uncertainty factors into account, we are left with the concentration in microgram per meter cubed. The TI is then further calculated into the TE, tolerable exposure. Since we are dealing with a gas pathway device, we will use the inhalation rate multiplied by the TI, ultimately being multiplied by the UTF, which can be adjusted to get a tolerable exposure in microgram per day. Once we have the tolerable exposure, the margin of safety is a fairly easy calculation, as we were just looking at the ratio of the tolerable exposure to the chemical constituent seen. Let's walk through an actual example. This example, we will look at dichloryl difluoromethane. This is a pretty common chemical, and it actually has a provisional reference concentration set forth by the US EPA. So for our point of departure, we are going to use that value of 100 microgram per meter cubed. Since reference concentrations are studied quite a bit, these values are robust, and they already take into account the uncertainty factors within. So we can just use a UF1 of 1, a UF2 of 1, and we can also use a UF3 of 1. However, this is a provisional RFC, and I'm going to be overly conservative. I'm going to use a UF3 of 3 as that conservative approach. Taking the point of departure and dividing it by the uncertainty factors, we then get a TI of 33.3 microgram per meter cubed per day. Now we must take the tolerable intake and turn it into a tolerable exposure. We multiply the TI by the breathing volume. Here I'm going to use the breathing volume of a pediatric patient, 10 meters cubed per day. And then we multiply it by the CEF of 1 and are left with a TE of 333.3 microgram per day. Let's assume that our chemistry report detected 26.7 micrograms of dichloryl difluoromethane. We then divide the TE by the amount seen and are left with a margin of safety. In this case, we have a margin of safety of 12.5. Since this value is above 1, there's a very low toxicological hazard. And furthermore, this margin of safety is also above 10, even with an overly conservative approach. Let's get into some appropriate risk mitigation, the top of the pyramid and maybe even the bread and butter for a toxicologist. Today, we'll be looking at scaling data. Scaling data always comes with a regulatory risk. However, most times, scaling data is appropriate if done correctly. We apply various scaling factors based on how the device was tested or how the device may be used in a clinical setting. The most common type of scaling is a surface area scaling. If the material is homogeneous throughout, the same inside and out, this may be the most appropriate scaling based on the patient contact portion only. For example, a breathing tube that is tested by air going through and around would yield results for the whole device, not just the interior of the breathing tube. For easy math, let's just say the scaling factor would be 50%, meaning that if a chemical constituent seen was 1,000 microgram per device, this would be scaled to 500 microgram per device. If a larger device, maybe a family representative device, was tested, but a smaller device may be used by a patient, say 70% of that, we could apply a scaling factor lowering that clinical exposure. Let's also say certain aspects of the device were tested. However, they may not ever come into contact with the patient. We could think of a cannula that a patient can breathe air in from one side and then expel air on the other side, meaning only one side of the cannula will be used for breathing. However, the whole device was tested. The non-patient exposure may only account for a small percentage. We will just say 15% is the non-patient contacting, making our scaling factor 85%, just for easy math. This will lower that exposure seen to 850 microgram per device. Condensate scaling for extractable and leachables is almost always clinically relevant. If a gas pathway device is extracted in, say, 1.5 liters of extraction fluid, there's really no way a patient may be exposed to that much in a single day, let alone every day for the device use lifetime. So we must determine a relevant amount of condensate to scale this down by. Most of the time, this is done by the sponsor. They weigh the device before use, run the device for as long as it may be ran in a clinical setting, or they'll run it for 24 hours, and then they weigh the device after. The weight difference is the amount of condensate that can be formed in that clinical setting. This is because the density of water is 1, so therefore 1 gram equals 1 mil. If the device had a 15 gram difference, it may yield a 15 milliliter condensate value. If we continue to use the extraction example of 1.5 liters and the relevant 15 mil condensate value we came up with, we will have a scaling factor of 0.15, meaning that if 1,000 microgram of a chemical was seen, the patient may only be exposed to 15 microgram per day now. 15 mils of condensate, that's still an awful lot to be exposed to. But once again, each device is different, and this is up to the sponsor to determine. Lastly, we can use a clinical device use as a scaling factor. Since exposure to the chemical is calculated based on the breathing volume per day, we can scale that exposure based on how long it may be in use. If the device is only in use for 12 hours, we can then scale the amount of chemicals reaching the patient by 50%. Or if the device is used 6 hours, we can scale down by 75%. Now, scaling doesn't have to apply to every single chemical. As a conservative approach, you can only scale the problemsome chemicals and leave everything else unscaled. Let's look at a real quick case study for, let's just say, a CPAP tube. Consider, for example, a tube that carries air to the patient at a maximum flow rate of 20 liters per minute. This has a minimum flow rate of 4 liters per minute, and the air is no hotter than 37 C. This device will be used in adults only for a maximum of 10 hours per day for greater or equal to 10 years. The device was then tested for VOCs at 0.1 liters per minute at 37 C over two 24-hour periods. Then particulates were tested at 50 liters per minute. There were no inorganics tested. Additionally, the tubing was tested by air being pushed through and around the device, meaning a greater surface area was tested. When comparing the clinical use to the applicable testing, this testing was robust and appropriate. This will yield relevant information and data to be used within a toxicological risk assessment. For the analytes seen coming off of this case study, we have the usual compounds: acetone, chloromethane, methylene chloride, dichloryl difluoromethane, and 2-butanone. We can therefore look at the calculations for each patient population and how they may be exposed to per day. Once again, we are only going to look at adults, as this device was only used in adults, and the TTC of 40 should be considered. However, there is relevant toxicological data for all of these compounds, so we will derive a TE and MOS respectively. Given these 5 compounds, all of the applicable margin of safety values, they will result in a value above 1, and we will not necessarily examine them further. Even though all these compounds have that favorable margin of safety, what if one of them didn't? This is where appropriate risk mitigation will come into play. Let's look at the same 5 compounds for the adult exposure and see how we could appropriately scale these down. In this case study, the device was tested through and around. Let's just say, once again, the homogeneous material was split perfectly by surface area, 50% on the inside and 50% on the outside. This results in a scaling factor of 0.50. Applying this straight down, you can just see the exposure gets cut in half. Another approach we can take is the clinical use duration. This device has a maximum use time of 10 hours a day. Well, the exposure for the adult population was based off of a 24-hour exposure. We can take the 10 hours and divide it by the 24 hours to get a scaling factor of 0.417. This scales down the compounds a bit more. Well, if you still have a problemsome compound using either or, you can apply both scaling factors at one time. You can apply the 0.5 factor for the surface area and the 0.417 for the clinical use to come up with an overall scaling factor of 0.208. While none of these compounds actually needed scaling, this was just an example of how to scale using the data at hand. Keep in mind that every time scaling occurs, it does have an increase of regulatory risk. There's also a few additional considerations that must be kept in mind. Further analytical testing may be needed to help demonstrate safety of the device. If there is a material change or a manufacturing process change in your device from what has been tested previously, additional testing may be needed on that device. If any components touch the skin of the patients, it could be a dual classification for that device as an indirect breathing gas pathway, as well as direct contact as surface device for the skin. A great example of this would be like a CPAP mask that fits on the face of a patient. This must undergo the VOCs and particulates, as well as the big three: cytotoxicity, irritation, and skin sensitization, as these three endpoints are not covered within the toxicological risk assessment. I want to thank each and every one of you for attending this webinar, and please keep in mind that Nelson Laboratories has much, much more than just your toxicological needs. We also have experts that specialize in packaging, sterilization, and biocompatibility, in addition to the hundreds of laboratory tests. For the Q&A section, I would like to introduce our board-certified principal toxicologist, Dr. Sarah Campbell, who may be chiming in to help answer any questions. Additionally, feel free to send me an email if you think of a question after the fact. Thank you.
Great. Well, thank you, Nathan, for that thorough and informative presentation. Once again, don't forget this presentation will be archived on mpomag.com for a year. Now, as Nathan indicated, we have time for the Q&A session. We'll try to get to as many questions as possible. Nathan, your first question. This is a little bit of a long one. For the application of lubricant jelly on a medical device, it's applied to the outside of a supragalactic airway mask head prior to insertion into the patient's throat. Would you agree with documenting the contact type as surface contact, mucosal membrane, humidified gas pathway, and externally communicating via patient airway?
Hi, this is Dr. Campbell. I'm happy to answer that question. So your particular device in question does fit into multiple different categorizations per ISO 10993 and also qualifies for categorization per ISO 18562. So those parts that are contacting the mouth would be considered that surface device mucosal membrane contact. For the inner airway, that would be your air VOC, and for your condensate that could be collected within the interior of that device, you would also need to conduct some sort of extractable or leachable study. You are also, based on this type of device, an externally communicating tissue contact device, so would be required per ISO 10993 to address the biological endpoints for that categorization as well.
Okay,
Thank you,
great.
Sarah.
Thank you. For the next question, in your opinion, would an endotracheal tube used in intensive care represent a gas breathing pathway which produces condensate? Consider the condensate produced inside the tube running down through the tube and into the patient, alternatively being sucked up if the tracheal tube has a suction function. Would love to hear your thoughts on this. Thanks.
Yeah, this is kind of a special case that we can look at as well. I guess this is very case by case, and I guess the answer to this question will be it depends. The condensate produced, it does have the opportunity to be introduced to the patient. However, if the tracheal tube does have that suction function, is there a possibility that some could leak out, or is there a possibility of that suction function not working correctly, say less than 1% of the time? To cover all your bases, it may be best just to do that ENL testing on the interior of that as well.
Okay. The next question we have, does the sampling take place in a controlled environment?
Yeah, the sampling for the gas pathway devices, they take, the sampling is ran in a glass chamber. It has really no ability to get contaminated by the laboratory environment. It's not ran in a clean room. It's just in a glass chamber where it's the device only, and it's all in a closed circuit from the air flowing through from the beginning to the very end in the sampling chamber.
Okay. Another question, Nate. You said that air could be tested through and around the device. Is there a preference?
Really, we like to run the preference of how the device is actually used. We want to have that clinically relevant testing. However, as a more worst case, going through and around can be seen. But if the option is there just to run the testing through the device, that is a good option.
Okay. I apologize if I'm not reading this exactly correctly, but I think you'll follow it. When calculating the TE equals TI by breathing rate, if TI is available in mg per day, how to calculate TE since breathing rate is available in mg/m3?
I'm happy to take this one. So you can find points of departures, these toxicological thresholds in the literature for animals exposed to a concentration of the chemical or animals actually dosed, say, orally with the chemical. So in those cases, you would, if it was dosed in air, you're going to get your milligram per meter cubed concentration. The end result is to get that TE in some sort of microgram per day threshold. And so you'd be doing the correct conversions based on your patient population. So for those that are, the points of departure are in milligram per kilogram, you would be using the intended patient body weights or the default body weights given in ISO 10993 part 17. So essentially, you're always going to get down to whether you have a concentration given in microgram per meter cube for a chemical or milligram per kilogram, your TE will be in microgram per day, and that will be what is used to calculate your margin of safety.
Okay, thanks. Sarah, I have another one for you. What are your recommendations in assessment or anesthesia devices to cover for the exposure to anesthesia in the gas?
Yes, great question. So it's definitely not to test with the anesthesia. These anesthesia gases show up in our chromatograms, and they essentially are very large peaks and inhibit our ability to see any chemical that is co-eluting with the anesthetic. So it would be air VOC testing without the anesthetic. And you can see some notified bodies wanting some sort of extractable or leachable, but I would say it's inappropriate. Those anesthetic gases by themselves are nonpolar. The air VOC testing, either done in air or pure nitrogen, is nonpolar. So we rationalize the air VOC testing is the only thing necessary when the anesthetic is given in dry air. So if it's going to be given over a long period of time with humidified air, then you need to start thinking about doing some sort of extractable or leachable per ISO 10993 part 18 with solvents that represent the polarity of whatever anesthetic or nebulized gas it could come in contact with.
Fantastic. We have another question. If our gas pathway is made of PC material, can we get inhalation limit in any literature or any website?
Yeah, you could derive kind of some inhalation limits based on literature for the material. You might have to look at everything that makes up the material, and it's not going to be as specific as, it's not going to be as black and white. It might be kind of more gray. But literature searches will be great. There's some databases that may or may not have some limits using that exact material. I would suggest kind of going through some literature, looking at relevant toxicological studies, seeing how relevant it is to that material, and then just seeing if there's any exposure limits for that.
Okay, another question. Asking, how often do you get regulatory questions and how does Nelson Labs handle them?
Yeah, we get regulatory questions every so often. Sometimes they're more of clarifications. Some range from how the test article was set up in the sampling chamber to specifics about the toxicological risk assessment. Each one of our toxicological risk assessments comes with a certain amount of regulatory support, which we then use to answer or provide that clarification to regulators based on the sponsor's behalf.
Okay, fantastic. Well, unfortunately, that is all the time we have for today's presentation. I'd like to thank our presenter, Nathan Frederick, for his insights. I'd also like to thank Sarah Campbell for her analysis during Q&A. And I'd like to thank our sponsor, Nelson Labs, for its support. Finally, I'd like to thank you, the viewer, for attending this session. I hope you found it to be a valuable experience.